Hi, welcome to part two of our discussion of neuroanatomy and networks in the brain. This is still Howie Rosen, and let's move right on to the discussion in part two. So here again is the overview for part two. We're going to discuss more ways of studying networks in the brain and the implications of those kinds of techniques and analyses for understanding the brain. So in the last session, near the end, we talked about different ways of studying brain activity using imaging of blood flow with PET scanning and with MRI. And so all of those kinds of analyses that do bring out the activities in networks, they use the idea that we can image the brain during an activity and then image the brain during a control activity and compare those images to understand a network, which is more active during the task than during the control task. In the sort of 2000s and after that, we discovered that there's another relationship within networks that can also be exploited to understand the networks, and this is the variation in blood flow at rest. So it turns out that what I showed in the first slide, which is that blood flow increases and decreases in response to the task, that there's also kind of varying blood flow over time in different regions of the brain, and not all regions have the same exact pattern of when blood flow is going up and when it's going down, that there are slight differences across different regions in the sort of normal up and down rate of blood flow. And it turns out that regions that are part of a network, that are connected with each other, have more synchrony when their blood flow is going up and down, even if you're not doing anything, than regions that are not part of a network. So essentially, the way we discovered that is by looking at these relationships in the following way. This is at least one way, and this is one of the early ways. Let's say you're interested in this little red spot, part of the brain. This is the superior, it's the middle frontal gyrus on the right side of the brain, and you're interested in what networks that middle frontal gyrus is part of. So you can put a seed in a functional MRI image, set of images, and then say, okay, let's look at the blood flow in that region over time by looking at the variation in MRI signal over that time. And if you do that, that produces a time series where you see the blood flow over time is going up and going down and going up and going down. Now you say, well, are there other regions of the brain that are correlated, that have the same pattern of blood flow up and down? So what you do is you say, I just looked at that brain. Now I'm going to look at the time series for every other region in the brain that I'm interested in. I've only got a subset of here, but you can literally look at the time series of every voxel, every 2 square millimeter part of the brain, and ask, what are their time series of blood flow? And so, for example, if we take this region and we say, well, what's the time series of blood flow there? And you see, yeah, there's an up and down and up and down. But when you compare, they're not matched very well with the one we were interested in, the one in the middle frontal gyrus. So when the middle frontal gyrus blood flow seems to be increased, actually the one in that inferior frontal gyrus that I was showing you is actually decreased. They're almost opposite. So that's not a great correlated region. But if you look at this other region over here, which is the sort of superior parietal region, its blood flow matches the blood flow in the region that we're interested in pretty well. So then what you decide is those two regions are in a network with each other. Now I blocked out the other those other three regions that are now in gray, assuming their blood flows did not correlate well with that middle frontal gyrus region either. But this is the principle that we're using. The way we described it, we found two regions that we believe are now in a network, and we found at least one other region that's not in that. So what we could do is study networks using that approach, taking regions that we're interested in and seeing what regions are part of its network using this resting state blood flow analysis. And what's been found is that you can develop networks, and in fact, they make a lot of sense. So here's an analysis on the bottom, on the left side of the slide, where they looked at a resting state network that was relevant to language using a seed or a similar approach that looks at language-related networks. And they asked, well, how similar to that is the network that you would get if you did a task that activates regions that relate to language? And you could see that, the regions of the brain that are, bright yellow and red, they're almost the same exact regions as come up in a task-related fMRI from a language task. So it really highlights the fact that these two different ways of looking at blood flow, one during activity and another during rest, they really revealed potentially the same networks just using these different approaches. I'm highlighting a language network on the left, but what we found is that you can identify lots of different networks that make a lot of sense using these approaches. So if you seed a region in the motor cortex, you could see other regions of the brain that have to do with physical functions. If you seed a visual part of the network, you find other regions that have to do with visual processing. If you seed a memory region of the brain, you see a memory-related network, etc., etc. So this resting state activity has become, a very prominent way of studying the brain because it's for many reasons, but frankly, one reason is it's much easier to do than the activation studies. You don't have to have screens in front of people. You don't have to have them doing anything. You just have to have them rest in the scanner when they're getting a functional activity study. There are other useful reasons to use it, but that's a really important one. So now we're finished discussing the way that we study networks with functional activity. And now we're going to talk a little bit about an approach that's been used to look at the actual connections between different regions of the brain. So in the first half of the talk, I showed that using dissection, you could see these fiber paths, fiber bundles that connect different parts of the brain. Now I'm going to talk a little bit about a method that can image those fiber bundles using MRI. So, essentially the technique is based on the movement of water in the brain. So when you do an MRI, you can image lots of different chemicals in the brain, but all the MRIs that you usually see every day, they're really imaging water because they're looking at the signal from protons, and the most common molecule in the brain that has protons in it is water. So when you're MRIing the brain, you're MRIing where the water is and what the water's environment is. And one of the things that people know is if you have a molecule of water, and you have many molecules of water, they'll move around. And if you set up your MRI so that you can kind of label water at one time and then trace where it's gone after a few hundredths of a second, then you can get an image that tells you about water movement. And the direction and intensity of water movement can tell you something about the environment it's in. So if the water is in an environment where it can move in any direction it wants, then basically if you image many water molecules in that region, you're going to see random motion and going all different directions. And the likelihood of water moving is equal in any direction. So that's called isotropic diffusion. Water is diffusing from its initial location to another location in a sort of random fashion where it can go any direction from where it started. And it's equal in all directions. It's equally likely to go in any direction. So it's pretty unconstrained is what you're learning from that kind of analysis. If the water is constrained so that it's, for instance, in a structure where maybe on two sides it can't move in that direction because, for instance, there's fat in the way and water doesn't want to interact with fat, then it can't move in those directions as much, but maybe it can move up and down. That's called anisotropic diffusion. In other words, it's not even, and in fact, it's more intense and likely that water can move in certain directions than others. By imaging the brain, you can start to see water molecules that might have isotropic diffusion and I'd see other molecules that move in an anisotropic way, that are constrained in how they move. And so it turns out that if you look at a neuron, a nerve cell, and you look at its axon, when a nerve cell sends out a major branch called an axon from it, that branch has membranes, and the membranes are often surrounded by myelin, which is a fatty substance. So essentially, that axon is a tube that really prevents the water inside it from moving in all the directions it wants. It turns out that the water can move very nicely up and down along the axon, but it can't move as well out, across the axon or out away from the axon. So the movement within axons is anisotropic. And essentially, by imaging, which directions water is moving, and how intensely, you can get estimates of the size and the integrity of white matter tracks that we were talking about earlier in the first talk within the brain. And so that's essentially what this kind of imaging do. We call this diffusion weighted MRI. In other words, it's MRI that is designed to detect the strength and the direction of diffusion of water within the brain. And I just really have this image here for something to look at, so it's not all words on the slide, but this image is from a website which depicts the sort of standard approach for how the MRI, for diffusion-weighted MRIs are collected and processed to create maps at every region of the brain of what the main direction of water diffusion is in that part of the brain, and you could assemble that all into tracks. I've highlighted here on the bottom, within the Canvas website, there are optional videos and papers that you can read discussing the details of this kind of technique and more if you're interested in that. But just to kind of show the point that we're making here in the talk, once you have these methods, you can then create images of the tracks you're interested in. And here, for example, on the left is an image in one person of the corpus callosum, that major tract we talked about earlier that connects the right and left hemispheres. Here's in the middle, so the corpus callosum's on the left. In the middle of the slide, you have another tract that's identified, which is the arcuate fasciculus. This is one of those tracts we showed earlier that you could see in a dissection of the brain that connects the temporal and parietal and eventually the frontal lobes as well. It's that big C-shaped yellow tract. And on the right is another tract called the uncinate fasciculus that connects the frontal and the anterior temporal lobes, shown in purple. So these are the kinds of images and analyses you could get from this diffusion-weighted MRI. You can obviously, hopefully, see the relevance to network analyses, because now, in addition to be able to see the activity and blood flow in different regions of the brain, you can actually image the fibers that connect them. So, we've talked about the different methods for studying these connections and networks within the brain. And the question is, why do we care about these networks and how can we use them? Well, I think broadly speaking, there are at least three different ways that people have tried to utilize these techniques. One is for neuroscience, trying to understand what regions participate in a network and what each participant in that network, what its function is, and how the network as a whole functions, based on the sum of all those parts. And that's just to understand normal brain functioning. But other ways we've tried to use this is to see how injuries and other kinds of damage in the brain might perturb the networks and see how the network responds. This can both tell us how the network works and also might tell us a bit about disease and about potential interventions. And lastly, it turns out that these networks are also help a part of how we understand our vulnerability to disease. In the next few slides, we'll talk about all these concepts a little bit. So the first one is neuroscience, and I'm not really going to talk very much about this. There are many, many papers looking at this, and this is one that I pulled out, just a nice example, that talks about all the interconnected regions that are part of the language network, and tries to, through the analysis of the fiber tracks and activation data that we have, tries to say what are all the regions that are part of the language-related network, and what are the potential functions of each of those regions. And this is based on the accumulated evidence from lots of activation studies and connection studies. This is just one example of this kind of work. You can read this paper if you like. But I'm going to also go on to the brain injury concept and how this work has been thought of as contributing to that. This actually is a paper from 2001 or 2, I think, where the idea was to look at how the brain responds to a stroke that causes an aphasia. And in this study, we actually studied 6 patients with aphasia, all of whom had lesions in the sort of Broca's area. And we know that that's an area that activates during complex language tasks like that verb generation study. But now that region can't activate because it's been injured. So what we were able to show is that actually when that part of the brain is injured, you get more activity on the right hemisphere, the exact homologous area on the right, during that same verb generation task. So this is an example that we'll utilize to sort of think about, well, what are the implications of that kind of finding for how you might treat patients with brain injury in that region? And we'll use this as an example. And many other examples of this kind of studies in the literature. Another way that that networks have been used is to understand vulnerability to neurodegenerative disease. So, one of the things we've observed over the years is that each neurodegenerative disease is associated with a pattern of specific regions of the brain that, even across different patients always seem to be reliably involved and that produce a recognizable set of symptoms. So in Alzheimer's disease, we have, memory and visuospatial deficits. And you have reliable injuries in the hippocampus and the parietal lobes, for example. In the behavioral variant of frontotemporal dementia, you have injuries to the insula and the anterior and inferior frontal cortex, and those patients have social and emotional behaviors, et cetera, et cetera. We have examples here in this slide for several different neurodegenerative syndromes and the patterns of brain atrophy that are typical, shown in blue on this top slide. And this is a paper from Bill Seeley from the mid-2000s, where he said, well, what is the basis of this kind of recognizable pattern of brain atrophy? What does it mean about neurodegenerative disease? And so one way he asked that is, well, these regions that seem to be involved, they're part of recognizable networks that we've heard about in the past. So, for instance, the parietal lobes, as I mentioned, on the right, they're important for visuospatial function. On the left, they're important for language function. The regions involved in the behavioral variant, we know from neuroscience those are important regions for social and emotional processing, et cetera, et cetera. So what Bill said is, well, how closely do these patterns of degeneration actually correlate with what we know about networks? And he said, well, if an atrophy pattern starts in a certain part of the brain, then if it's going to be related to atrophy in other parts of a network, we ought to be able to see that. So he took each of these syndromes and he said, what's the worst part of the atrophy in that syndrome? Where is it that all patients have atrophy and it's the most severe? And so for each syndrome, he identified where that region was. They're highlighted in circles on each of the syndromes. And he said, let's make that a seed for a functional MRI, a resting state fMRI experiment. And so he said, I'm going to take each of those regions and make it the region of interest to define what networks is it a part of based on resting state metabolism, which we talked about at the beginning of this talk. And what he found is, if you look at the seed for Alzheimer's, all the regions connected in the network basically recapitulate the pattern of atrophy that you see in Alzheimer's disease. By the way, just to be clear, so that's depicted in the yellow maps in the middle of this slide, in the middle of the image. And so this is all done in normal. So you take the seed from the atrophy pattern, the worst region of atrophy in that syndrome, and you make it a seed in normal volunteers, and you study their blood flow. So essentially what he's saying is the pattern of atrophy that you see in Alzheimer's matches the functional activation network using resting state MRI that you get in normal people if you use the worst region of atrophy in Alzheimer's as your seed. And he basically replicated that in each of the major neurodegenerative syndromes where the relevant atrophy pattern exactly matches the corresponding network of blood flow relationships in normal individuals. And even found in normal individuals, as shown in the bottom, that brain volumes in normals across these regions were also correlated. So if you're interested in the right parietal lobe, it turns out that the other regions of the brain that are part of that network based on resting state also are more correlated in terms of their brain volume. So this was really the idea that said it really reinforces that the atrophy patterns in neurodegenerative disease are a reflection of the normal networks in the brain and the membership of different regions of the brain. And it's really it's really led to the idea that maybe some of the pathological actors in these neurodegenerative diseases actually might move from region to region through networks in the brain. So now we're going back to an image made from diffusion imaging, and you could see that there are frontal regions like this uncinate fasciculus shown in red. The connections within that uncinate fasciculus are a potential vehicle for transfer of toxic proteins, for example, between different parts of the network that degenerates in frontotemporal degeneration. And you could kind of justify that same kind of logic with the other networks and how they relate to other diseases. So we haven't really proven this idea in humans that neurodegenerative diseases spread through these networks using the established connections through the white matter, but there is some evidence of it, and it's an ongoing area of discussion. So this is the end of part 2. Hopefully through viewing these 2 parts, you can really appreciate these take-home points, that there are specialized regions within the brain, but that even though there's specialized regions, they're interconnected with each other in larger networks that can be studied with various approaches: functional activation studies, resting state activity studies, and through studies of the white matter connections between them. And that can be useful for these various purposes, including studying the organization of the brain, thinking about how the networks might be capitalized or understanding networks for treatment. And we'll talk more about that in the live session, and that they're a potential basis for understanding the spread of disease through the brain. Thanks for listening, and looking forward to talking with you about this.